Hello, everybody. My name is Diana Phillips. I am currently a graduating senior in marine biology, and I do deep sea jellyfish research utilizing R. So I'm going to be teaching you today the introduction to R for research. So starting off today, before we get into anything else, let me show you where the resources that you'll need for today are located in your Canvas page. So this Canvas page is exclusively available for marine biology and environmental science majors. So if you're any other major, you're just going to need to email Mr. Lorenzo, and he will give you all of the resources that you need for this workshop. So the first thing that you're going to do is you're going to click on the NSU Marine and Environmental Science Careers Resource Hub, and you're going to scroll down until you get to past workshops. So there you're going to see Introduction to R for Research. This is our flyer. Then underneath, you're going to see our download instructions. For our workshops, we utilize computers in the library, which already have R downloaded. So if you're planning on doing this on a personal laptop, you can utilize these instructions. Here's the PowerPoint that I'm going to be going through today. Underneath, this is a practice CSV document that we'll be needing for one of our lessons. And then here is a step-by-step guide of everything that you're going to be doing in this workshop. So I highly recommend that you open up your step-by-step guide as the video is playing so that you can practice these skills as I discuss them. So let's head over back to our PowerPoint. So what is R? R is a statistical computing software. It allows us to clean and analyze large data sets really easily. For instance, with my jellyfish research, I utilize tons of these resources. And so I have over several million data points, and so being able to put it in R and having a software that can actually compute it is really important because, for instance, some of my data sets are so big that if I try throwing them in Excel, they won't even open. And even when I use more powerful softwares offered by Microsoft, I still run into issues where it crashes or there's just an issue with it or something along those lines. So being able to actually import all of my data, even if it is a really big data set, and then having it be able to clean it is really, really helpful when it comes to large scale research. The next thing is that it reproduces your results consistently. This is important when we talk about things like GIS, where sometimes the analysis that it does can be really inconsistent, and you might do the same thing a couple of times and you're going to receive different results. The thing that's awesome about R is that even if you're asking it to randomly select something for a particular reason, you can actually seed it to be the same random every time. So using a particular seed, you can get it to do the same random every time. So it's still random, but it's consistently random. And so that means that no matter how many times you do it, you're always going to get the same result because you have used the same random to get your result. And beyond that, whatever you're doing, it reproduces results very consistently. One thing to keep in mind with that is that if it's reproducing inconsistent results, that probably indicates that you have an issue with your variables somewhere. And so it's something to keep in mind that if you're receiving inconsistent results, the software isn't reading something correctly. Next, it is also cross-platform compatible, so that means that whether you are using a Mac or whether you are using a PC, it is going to be functional and you'll be able to utilize all of the packages. The only thing to keep in mind is that there are slight changes in the way that you input whatever it is that you're trying to do. And so it's just important that if you're utilizing a Mac, just be aware that there might be some slight changes. But other than that, everything else is compatible and will work. Thousands of free packages are available and accomplish a wide array of figures and analysis. So if there's anything that you've ever wanted to do in analysis, if there's anything that you've wanted to try, likelihood is somebody has already built a package for it in R. So for instance, I use a pretty uncommon variable for marine biology, which is a variable called spiciness. And there's actually an ocean package that has that very specific analysis that I need to do already inside of it. And so that's like a really cool benefit to me as an up and coming researcher, is that I don't have to code a formula by myself. I can just use a package that already has all of those details pre-coded. Next is that it's industry standard in marine biology for publication level research. So if you're hoping to go into grad school, R is going to be really essential. It's actually required to have a fundamental understanding of R if you're hoping to go to graduate school at NSU. So not only is it like this very powerful, very helpful resource, but it's also really required in your future field if you hope to go into research. So now we're going to talk a little bit about R and RStudio. So R is the actual software that you're using. This is what you need to do in order to accomplish anything. And then RStudio is a user-friendly interface. So it's important to remember that you need R to be downloaded in order to use RStudio, because RStudio is just making it easier to do a bunch of different things at the same time and to bounce between your resources, whereas R is the actual like fundamental software that you need. So what does RStudio look like? Here's a breakdown of RStudio. So at your top left, this location is where you're going to write and save your code. So here, just know that you can erase your code, you can fix your code, you can alter your code, and something else that you can do because you can save your code is that you can use these little hashtags that you see in green. And those hashtags are actually notes that the author is writing to themselves. And so it allows you to organize your code in case you need to remind yourself of potentially what you're doing or what you're hoping to accomplish. Just know that maybe you can only see one of those two boxes at a time. So utilizing the little, here, let me use the annotation tool so you can see my mouse. But utilizing this little window allows you to either make this larger or smaller, so you can optimize the size of all of these. And you can even go into your settings and change the orientation of them if you would prefer them to be in different locations. So just keep in mind that if you're only seeing one of these two boxes, the likelihood is that one or the other of these needs to have their windows opened. Um, and that's not terribly important. It's just something to keep in mind, especially if you're trying to go back and erase some of your code and rewrite it. So then when we go to talk about our bottom left, here's where the code is not saved. So every time you close your computer or reopen RStudio, that code is going to be deleted. And then you also can't erase something and rewrite it. You have to rewrite the entire code. Or you can use shortcuts so that you can make your last code reappear and you can alter it. I know some people who don't like using the top left corner, they only like writing the bottom left and they just clean it as they go. I know I personally really like keeping track of everything that I've done and the different iterations of things that I've done. And I like writing a lot of notes to myself, so I prefer working at the top left. But that's just up to personal preference. No matter where you write the code that you're planning on doing, it is not really important. At the end of the day, both locations are going to get you your responses. So now when we talk about our top right, this is where you have access to your history. So you can view all of the past code that you've written, and you have access to your environment. So that allows you to see your objects, your vectors, your data sets, and your matrices. So you can really see everything that you have put into RStudio. And so if you reopen it again, oftentimes, unless you save your workspace, your environment will be deleted. And so it's a good way to double check if something's not working. Maybe it's just not in your environment and you need to reintroduce it into your environment. So for your bottom right, this is where you can access all of your computer files. It opens exactly the same way as your files folder in your normal computer. The only thing to keep in mind is that you can't search for files by name. So just know that that means that you need to keep it really well organized. You can also use the plots in order to view any plots and figures that you create. You can use the packages tab in order to enable and disable any packages. And you can use help to access R's resources. So for instance, if you're opening a new package and you don't really know all of the things that are required for you to code in order to make this package work, you can open up the help and then you can actually read what you need as you're coding it in, which can make it really helpful, especially as you try new things. So first, we're going to start with our first lesson. So if you're working through the handout, you will see this as your first lesson in the handout. So we're actually going to start in R instead of RStudio. And this is just to show you that you can accomplish everything in RStudio you can also do in R. So we're going to start by typing 3 plus 4, and then In order to make this work, we call that running a line. So in order to run a line, you're going to press Control and you're going to while you press Enter. So you're going to write 3 plus 4 and you're going to press Control Enter. And you should receive a result. So something to keep in mind is that it's giving you 2 numbers. It's giving you the answer to your question, which is 7, and then it's also giving you a 1 value. So 1 means that it's currently showing you like 1 line of um, responses. So for instance, when I work with 999 variables, oftentimes every line will have that different number inside of brackets. And then additionally, at the end, if I have more than it's going to show me, at the end it will give me brackets with all of the leftover numbers of variables that I didn't get to view. So you just ran a very simple function with R. So this is just utilizing the base information that comes with R, the base systems that are available to you. So what can I do with basic R graphics? So you can create box plots, mosaic plots, dot charts, line charts, scatter plots, pie charts, histograms. You can do all of these things before you download a single package. So R by itself is really powerful before you even start using some of the other resources that are available to you. So let's try it out. So now you're going to open up that search bar again, and you are going to open R Studio this time. So you can see that the difference between R and R Studio is that R only has 1 window, and here you have your 4 windows available. So now you're going to type in these vectors. Obviously, since you're watching a video instead of me being able to help you in person, one thing to keep in mind is that it is really important to get your cases correct. So you want to make sure that you are typing in a lowercase a lowercase b, and a lowercase c. So what are we actually doing here? So A is the name of the object that we're creating. This little signal that looks like an arrow, which is made of a, I believe that's a less than sign, and then also a dash. That is attributing your object. So you are saying A is the name of my object, and C, I think of it is count. So this is how you can create a vector. So a vector is just a series of numbers. And so you are creating a vector by giving it a C, and then you're putting all of the numbers that you would like to create your vector with. So you're saying A is my object, and here is the vector that's assigned to that object. So and just keep in mind, these are random numbers that I created, and the thing that's important is not what numbers you put in here, but what's important is that you have the same number of numbers in both your object A and your object B. So now we're going to run a line of code. We're going to start with plot, and then you're going to do inside of your parentheses, you're going to do A, and then the little squiggly between A and B is a tilde. So make sure that you're using a tilde and not a dash. And you can stop right there. You can actually run just plot A tilde B. That will get you this plot. So all of the additional stuff that we're adding to this is actually giving us all of the pretty stuff in our graph. So main stands for our main title, and we put whatever we want for our title inside of quotation marks, and we do this so that it knows that it's writing something on your plot. Now for XLab, that stands for our X label. So again, we put it inside of quotation marks so that you can see that we are labeling it B. For our Y lab, that stands for our Y label. So that is again in quotation marks, and that is A. So now we're going to try a different kind of plot. This is our box plot. So again, you can see that now you're doing a totally different kind of plot, and you only have to change a couple of letters. You only have to write box plot this time. And you do the same thing, A tilde B. And once again, this will totally run without any of the additional writing. The additional writing is just to add the pretty stuff to your figure. So great job. Now we're going to try something a little bit more complicated. So we're going to start with observations. So here you see that I'm using an equal sign instead of that little arrow that I created before. You can utilize both. Just keep in mind that if you use an equal sign, sometimes it can get your functions confused. So it's always better when you're attributing a variable to utilize that little arrow sign instead of the equal sign. But just know it is interchangeable, so you can utilize either. It just might come with problems if you're doing a more complicated code. So once again, we are naming our object. So our object is called observations. And once again, we are creating a vector. So we have a series of numbers associated with our vector. And then underneath, you see year twice, and you see it written in two different ways. So the first way that it's written here, we utilize 2001, and then we do semicolon 2005. So that means that it's going to go from 2001 to 2005. So a different way to write that is you can do it this way, where you can say that you're doing a sequence, and then you type in your two numbers, and you tell it that you want it to go by 1. So that means that you are going to go as many numbers between those as you can get as you move by 1. So that means that it's going to go 2001, 2002, 2003, 2004, 2005. So for instance, what's going to happen if instead of 1, I write 100 or 1000? Well, it's not going to come back with anything. It's not going to work, and it's not going to work because there's not 100 or 1000 numbers between 2001 and 2005. So that's one thing to keep in mind, is that sometimes you have to think logically about why something might not be working. Because for instance, if you're doing 100 or 1000, that's physically impossible for a sequence to occur between those numbers, because you only have 4 years between 2001 and 2005. So next we are typing in year and pressing enter. So when we type in year and we press enter, we can actually see down here that we have, again, that 1 inside of our brackets, which means that we are seeing one line. And we can see 2001, 2, 3, 4, and 5. So just like I was saying before, it is creating that sequence for us. So now we're actually going to plot this. So here we see that the first thing we're going to do is we're going to plot our object year. Next, we're writing N, and then we're going to do PCH. So PCH actually stands for the shape of the object. So what you can do is you can go into Google, we'll do it right now, and we will type in So we're going to do our shape of our objects. So when we do this, we are basically attributing a number, and that gives us all of these different shape options. So you can see that it basically goes from 0 to 25. And so each of those is a different object. And sometimes you can fill in the interior, sometimes you can't. It just depends on the kind of shape of object that you select. So now we're going to go back to our function. So the next thing is color. So color is going to be the outline. And then BG stands for background. So that will be the interior color. And that stands for, and so in this case, we're writing pink. I highly recommend after you write this line of code, mess around with it a little bit. There's so many colors that you can use in R. And you can also use the number codes if you happen to know them for colors. Those also work in R. And then your CEX stands for the size of the dots that you are making. So if you make a bigger number, it's going to be a bigger size dot. And if you make a smaller number, it'll be a smaller sized dot. So hopefully, when you ran this line of code, you're going to find that there is an error. You typed in an object called N, but N doesn't exist anywhere. So that means that you made a mistake, in this case, an intentional mistake. So the first thing that I want you to do is I want you to take that error code that you just received, and I want you to throw it into Google. This is a really important part of coding, being able to make a mistake and then put it in line and understand what the internet is telling you is wrong. So here, it says error, object N not found. So again, as we discussed, your object is observations and years. So when we created our plot, we used years, but we didn't put observations anywhere, which means that if the object N is not found, I need to put in an object that it can find, which is observations. So here's just a little bit about errors. Oh, and then after you fix that, it should work, and you should be able to see that plot pop up. So errors. Always completely read your error messages. One of the biggest things that I see with people who are new at R is they do not read their error messages, or they get lazy when they're reading and they do not read the entire message that appears. It is important to read the complete message because it's going to give you a better idea of what went wrong. Additionally, if you don't understand the error message just by reading it, then throw it into Google, and there will be a whole community of people who are going to help you. That's something that's really nice about having a free and open source software that anybody can download, is tons of people have problems with it, and so there's a huge community of people out there who will help you. And then if it's still not working, it's often just a silly mistake in your code. So all you have to do is review your code, and you should be able to catch the silly mistake. One silly mistake that occurs really, really often is that you have to make sure your uppercase and your lowercase are correct because spelling matters and your cases matter. So everything has to be spelled correctly. Another thing to keep in mind that occurred during our workshop is that somebody forgot to actually run their line of code, and so it wasn't working, and they're like, why isn't it working? I'm pressing enter. So they forgot that you have to press Control Enter in order to get that line of code to run. So just make sure that you are actually running every line and make sure that it is spelled correctly if it's not working. So what if I want to do more than is available in my normal basic R? You're going to need packages. So packages are bundles of tools that are available to download for free that give you tons of resources for all kinds of cool things, basically anything to your heart's content. So the first thing that we're going to do is install the package. Some people during the workshop skipped this step, so please make sure that you actually install the package before you continue going. So you're going to go install.packages. You're going to use your parentheses, and then you're going to use your quotation marks. So we're going to be using the package ggplot today. So the next step after you install it is that you have to enable it. So the way that we enable it is this library function. So you're going to do the same thing. And then if you go over to your packages tabs, you should see that after you write library, there should be a little check mark. And you can actually just go into packages and you can just put the check mark next to the package you want, but it's better to practice coding. So I highly recommend you practice coding. It's also faster if you can learn and remember how to do it. So the next thing that I want you to do is I want you to go into the help page and I want you to go into ggplot2 and I just want you to read through the help page and just see what's offered and just learn a little bit about the system as a whole. So this is a graphic system. So this is particularly created in order to plot things. So now we are going to practice running t-tests. So a lot of you have done t-tests in your biology classes, in your genetics classes, in your ecology classes. T-tests are very, very important. So the first thing that we're going to do is we're just going to write t.test, and we're going to put that in parentheses and do A, comma, B. So when you're doing this, you're saying, I want you to run a t-test on my object A and my object B. And you should see a result down here. And next, so next you're going to run a summary statistic. So this is going to give you your mean and your quartiles and your minimum and your maximum. And so then you can run that for individual variables, or you can also run that for both. So now is when we're going to be using that information from the CSV file that I told you about at the beginning that's available on the canvas. So one thing people ran into is that you do have to download a package that will allow you to import CSV files or Excel files. So just make sure to do that before you keep going. It'll pop up an error message basically saying that it can't read it, and then it'll tell you exactly what package to download. So continuing from there, you are going to find it in your history. I highly recommend just looking in your downloads folder. It'll be right there in your downloads folder, or you can move it to an easier folder to find. And then you're just going to click on it. So after you click on it, you should see it imported here. And you're going to see that there are 3 variables. There's dogs, cats, and lizards. And you're going to click import. After you do that, you should see that sign up over here. And if you press these little arrow buttons, you're actually going to see that you can reorder them according to lowest to highest or highest to lowest. And so you can practice playing with the little buttons. You can also filter out stuff if you'd like to. So from here, we are going to start with our actual analysis. So we're going to repeat everything that we did before, but this time we're just going to use different syntax. So we are going to be using the name of the document, which is practice_r_workshops. And then we're going to use this $, which indicates the column that we would like to use. So in this case, I would like to compare cats to lizards. And we're going to see that pop up here. And just like before, this information is going to be found on your step-by-step guide. And so take a look at that. It shows you how all of the syntax will look with all of your plots and all of the labels. So just run through that. So yeah, I would pause here, make sure that you run through those so that you can practice those skills. So what if I want to practice after the workshop? So you're going to need a package called Swirl. It is a learning platform in R that teaches you how to use it. Go through the lessons at your own pace. Let's practice. So first you're going to do the same thing. You're going to install the package. You're going to install Swirl. Then you're going to enable it utilizing the library function. And now you're going to use Swirl, and you can do resume.class equals to resume a specific class, or you can just use Swirl with the parentheses, and you can use nothing else inside of it, and then that will also work. And again, you should see it enabled in packages. And now you're going to start working through. So you'll see that Swirl will actually communicate with you and talk to you. So again, this is a really great place to pause the workshop and just work through that, or you can do it after the workshop. So what other resources are available? I highly recommend the NSU Oceanographic Library website. This website is really great. You can click on it here. I'll show you what it looks like. So this website has all of the information on learning about R. It has the essentials of R, a coding clinic, data wrangling in R. So it has all kinds of resources available for you to work through. There's also a LinkedIn Learning for R that's available to you as an NSU student. So I highly recommend that you take the time after this workshop to look at some of those resources so that you can gain a better understanding. And then also, if you're interested in anything else, if you're interested in MATLAB or Primer or SPSS, the NSU Oceanographic Campus Library has all of those available. So really fantastic. What other resources are there available if you would like to learn more after this workshop? So you can also go over to DataCamp. It's a lot like Khan Academy, but it's for coding, and the link is in this PowerPoint. So you can actually go through, and there's courses available for you to practice stuff in R. Yeah. So with all of that being said, you can end it here, or you can continue to hear me talk about how I utilize this in my own research. So I will open up my research now. So I study deep sea jellyfish utilizing underwater robots, also known as the distributional patterns of mesopelagic Scyphozoa within the Monterey Submarine Canyon in California. I work with my advisor, Professor Feingold, and my research partner, Manu Ploner. So what is a scyphozoan? This is just a true jellyfish. And my research question was, how do environmental factors affect the spatial distribution of jellyfish in the deep sea? This research is really important because a lot of jellyfish get destroyed by fishing nets and stuff, so they're a really understudied taxonomic class. So I utilized ROVs from Monterey Bay Aquarium Research Institute, also known as MOMBARI, and we looked at the submarine canyon over in Monterey Bay, and we utilized their video annotation and referencing system. So this is just when they're going in the deep sea, they take a snapshot of an organism and they attribute temperature, water quality, all of this stuff to the sighting of the organism. And so I utilized that to identify observations of jellyfish over the course of 20 years. So I selected ones that were found in greater than 150 abundance over the course of 20 years. So we have Atolla, Periphylla, Parrelia, Stella Medusa, Deepsteria, and Paraphilopsis. So utilizing a different program called GIS, I was able to plot them spatially so that I could see where each of those genera were located within my survey area. And my survey area was 1 kilometer um, around the canyon. I also utilized two sites to be able to look for my spiciness. So more of the water quality side of this, I mentioned spiciness a little bit earlier. So as you know, when there's more stuff in the water, it's heavier, and when water's colder, it's heavier. So the results is what happens if you have low spiciness and a cool temperature. Well, you might have an area that has the same potential density as something that has high salt content and high temperature, but it's going to have a different variable called spiciness. So it allows us to see water masses and whether they're coming from polar or equatorial sources. And then it also allows us to view these distinctions using TS diagrams. So this is actually a TS diagram that I utilized in R. And so I actually created this using, I believe the plot function is ts.plot. And so you can see that I was able to replicate a very similar TS plot to what had been recorded in previous literature, which is really cool. And just keep in mind that this Atolla had, I believe, 15,000 data points or something along those lines. So I was able to plot an absolute ton of data in a matter of seconds. And it did not crash once. So just attributing to the power of this technology. So the other thing that I did in R is I utilized violin plots. So a violin plot is wider in areas with greater counts. And so that means that at particular temperatures, there was more observations at this temperature. So, from these results, we can see that Atolla, Periphylla, and Stella Medusa were located at higher temperatures than Deepsteria. And we can see that when we attribute this to depth, we see very similar findings, which is that these three tend to be located in shallower waters, which makes sense because temperature and depth covary. So when we look at salinity and oxygen, we can see that they're a lot closer in salinity and oxygen content. And so again, to the power of R, this is a really cool figure that you can make. And these little red lines inside were actually something that I customized. So if you would prefer to only have a dot where the mean is, or if you would prefer, these red lines stand for a standard deviation away from the red dot, so away from the mean. But you can customize it to only have the mean or to have nothing inside at all. It's just a matter of whatever you would personally like in your violin plot. And so customizability is something else that is really helpful. Here's something that I did not create in R, but you totally could. And the reason was the binning was a little bit awkward. But yeah, here I'm just creating oxygen figures, and I'm basically looking at whether my jellyfish are existing above the oxygen minimum zone or within the oxygen minimum zone. And you can see a really cool feature, which is that it very much resembles previous literature. So we can see that those jellyfish are having the same trends, even though we're doing this 10 years after the original survey. And then we are also looking at 20 years in time. So it's very cool to see the same trends replicated. That's what we want to see in literature. And we can see that with some of our other species, again, the power of R is that we can see very visually that like this is a species that caps off before you get to 400. And this again, also, you don't see it in depths of less than 400 meters. Whereas our Stella Medusa, you would probably find it in depths of less than 200 meters. But that is merely a result of the data that we collected. We removed anything under 200 meters. So they might have really high abundances in even shallower waters. So this is just the results from my study. This is not particularly to do with R anymore, but the results of my personal research is that we um, saw that Atolla, Stella Medusa inhabit similar depth, salinity, temperature, and oxygen ranges. So they had very similar environments, whereas Deepsteria and Paraphilopsis also inhabited similar salinity, temperature, and oxygen ranges. So our Periphylla and Stella Medusa occupied similar salinity ranges, but not any other ranges, and our Paralia had similar oxygen ranges to Deepsteria and Paraphilopsis. So this is something else that I did in R. This is called a Wilcox-Rank Sum test. It's something that we do when we don't have evenly distributed data. And basically, it's comparing each of the genera of jellyfish to each other. So we see here, for instance, that my Atolla and Deepsteria were occupying significantly different depths, which means that I got a super duper low p-value. Something to keep in mind here is that this E basically is like, it would be 2 times 10 to the negative 16. That's kind of how you can think of E. It's an abbreviation for the times 10 to the negative power. So we can see that everything that is not highlighted was significantly different from each other. And this is really cool because when we're doing tests like this has a lot of comparisons. Basically what it's doing is it's doing every possible mix of these genera and then it's comparing it to each other. So this is really cool because it's basically rearranging all of my data so that it can get the most accurate result. And I used an adjustment method called home, which is one that is not particularly strict. So if I wanted to make this super duper strict, I could use a different option. So that's, again, a testament to the customability of R, is that I can kind of change the way in which this happens. So we see that for our temperature ranges, once again, there's only one that is statistically insignificant. So the majority of these are occupying significantly different temperature ranges. Here we see 3 occupy very similar salinity ranges, and 4 occupy relatively similar oxygen ranges. So these are really cool results. They allow me as a scientist to compare these different genera with each other. And I can say definitively that these jellyfish are affected by their environmental constraints because they have been shown to statistically significantly not really coexist in the same environments. They're existing in different water quality measurements. And so the result of that is that we know that species like Atolla and Stella Medusa, which occupy the same depth ranges, salinity ranges, and oxygen ranges, are more likely to compete with each other than other genera which aren't. And that's really important because in these resource-limited environments, we want to know how the food web is working and how those ecosystem dynamics function. So this is a project that gives us greater insight to that. And I did the majority of the work for this project in R, which again is why it's, a standard in research that you publish. So for some unsolved challenges in this research, obviously we're only looking at one class of organisms, which is jellyfish, and there are tons of different classes of gelatinous invertebrates in the deep sea. So one of our greatest intentions was to gain greater insight into competition between these species and also into ecosystem functions that they may be having. So in future research, I would love to expand this to not just scyphozoans, but more classes of gelatinous invertebrates. And that's where programs like R can actually be really beneficial. Is this was a lot of data. This was millions of data points that I utilized in this first study. So especially if I intended to broaden my scope, I could potentially be working with even more genera of gelatinous zooplankton. So that's part of why working in R has really been able to level up my research and allow me to do such complex analysis that really wouldn't be possible in Excel. Yeah. So That is all I have for you today in our Introduction to R for Research workshop. I hope you guys learned a lot. I hope that you work in Swirl and that you broaden your skills, and I hope that you work in Data Camp and you can practice a lot of what you learned here today. So if you missed this workshop and you worked through this entire thing, just feel free to send me an email, actually. Um, And so if you send me an email proving that you went through all of your that you went through all of the lessons that I showed you here today, you can actually get a certification. So just feel free to shoot me an email at dp1773@mynsu.nova.edu, and I will send you a certification basically saying that you worked through the Introduction to R for Research workshop. So that just means that you were introduced to R. But yeah, so all you'll need in order to do that is send me an email saying that you worked through this virtual lecture and just send me proof of the code that you completed. Awesome. Well, thank you all so much for coming here today. I really appreciate it. And I hope that you learned something.